Okay, so hello, good evening, everyone. Welcome to today's section. Before we go ahead, first I'm going to start off with a quick recap of whatever we discussed last week. If we just follow the basic idea, we had continued with our idea of non-invasive diagnostics, where we had previously discussed cfDNA, CTCs, and the way they can be used for diagnosing cancer patients. Just yeah. So we had looked at that in the session previous to the last class, and after that, in the previous section, we had focused majorly on the idea of exosomes, what they are, what is the physiological need, what is their role in context of cancer diagnostics, and finally, we had ended on the discussion about the isolation protocols, which are used for separating these from the complete cell lysate. And we had just gotten an introduction into conventional cancer therapies. And today we will diverge away from this theme where we'll move more towards the recent advances which have been which are currently undergoing improvement and revision to make them more useful for the general public. We'll look at more advanced or more improved techniques that are being implemented for treatment of cancer patients. And some of it is something we have already discussed when we talked about the cell signaling mechanisms and all of the different cascades. And we discussed targeted therapy in that context a little bit. We had introduced, how you can target RTKs and how you can target all the different cell signaling, like the components within the cascade, so as to inhibit those pathways. So a little bit of that will also come into focus today because that actually overlaps with the current idea of looking at treating patients in a more specific manner, in a more targeted manner. To quickly brush up whatever was discussed in the previous class, we first understood what exosomes are. And of course, if you already have attended previous class, you would know that exosomes are basically cell-derived extracellular vesicles, which are endosomal in origin, which means that their membrane or the outer lipid bilayer is derived from endosomes, and the characteristic feature is their dimension, and they are technically the smallest type or the smallest variant of extracellular vesicles. Here we looked at the overall simplified process by which exosomes are generated. And you can see that it starts off with the process of endocytosis, formation of early endosomes, then fusion with lysosomes to form endolysosomes. From there, a large multivesicular body can be formed, which was also known as a late endosome. And derived from that, by the process of exocytosis, what is pushed out of the cell is actually known as the exosome. That is what we understood, the physiological way by which exosomes are formed. Now, under its definition, what you understood what exactly is contained within this particular structure. So that can include anything from proteins, lipids, mRNA, microRNAs, and DNA as well. They are the basic principles. Then we looked at the fact that they are generated as a result of a process whereby the cell membrane, it buds inward and then again it buds outward, and it sort of flushes out whatever vesicles have been generated. The key factor that we had focused on in the last class was the fact that exosomes are responsible for intercellular communication as well. So they're able to transfer or relay signals from one cell to another. Either it can be via interaction with the recipient cells or the second cell surface receptor, or it can be via some other interaction. It can be via transfer of macromolecules. A lot of different ways can come into play. Then we understood that under physiological condition, exosome biogenesis is a mechanism by which protein quality control is maintained, and it helps in maintaining the homeostasis between the amount of protein that needs to be synthesized and the amount of protein that needs to be degraded. And finally, we understood that it has critical roles to play in both human health and human disease. Moving ahead, we looked at the cargo within the exosomes in a lot more detail. So here you can see the same process like I just discussed. And here you can see all of the different types of cargoes which are possibly present, starting off with cell surface for transmembrane proteins like CD9. We had explained a little bit about what CD9, etc. are, or heat shock proteins like HSP60, 70, or 90, and integrins, some cytoskeletal proteins like actin or myosin, then RAP GTPase and Nexane, which are involved in trafficking across the membrane. Then we looked at just DNA in general, which can also be present, which is something we had discussed in the class before, where we talked about ctDNA being an important um, like an important diagnostic tool for patients. Um, Uh, then we looked at all the long non-coding RNA that can be present, circular RNA, the hypoxia associated or other different types of miRNA or other proteins like beta-catenin, cabulin 1, etc. Then we looked at yeah. Then we looked at the basic idea how they are relevant in context of patients who are actually undergoing cancer therapy in the clinic. So the fact that exosomal markers are reportedly some certain types of exosomal markers are reported to be much higher in a certain subset of patients being diagnosed with a certain type of cancer. Therefore, they're very attractive targets for detecting cancer within patients. So studies have actually analyzed the relationship between the amount of plasma exosomes and tumor burden. Also, we looked at certain examples of this, like for example, the cabulin 1 and CD63 positive exosomes to be found at much higher levels within patients who have poor prognosis for oral cancer. Then for prostate cancer, those with CD81 and high PSA were also used as a diagnostic tool. And also this approximately 10 microliter plasma sample was found to be enough to actually help in diagnosing patients with non-small cell lung carcinoma. So this was the simplified process which is followed for isolation of exosomes, which we have looked at in more detail later on. Then we understood the process of isolation. This was the more conventional route, which involved multiple centrifugation steps at different speeds for different durations, followed by filtering. And this is often known as differential or sequential differential centrifugation, where you can see in each sub-step a component of the entire um, media or the entire culture that you initially take that gets separated out, starting off with the cell pellet. Then large extracellular vesicles can be gotten rid of. Then from these small extracellular vesicles, what you finally end up with are only the exosomes. That is the consequence of a lot more ultracentrifugation. So this was the more traditional route that we discussed, and then we moved on to the idea of using affinity-based purification methods, which involved antibodies, therefore leading to the use of immunoaffinity isolation-based methods. So thereby we saw that antibodies were conjugated with certain beads. So let me just go to the next slide, which had a better schematic to explain that. So yeah, the first step is pretty much the same like we just discussed, whereby you use the conditioned media, or you can use patient blood, depending upon whatever your starting material may be. And then you subject it to sequential centrifugation steps and finally narrow down to only the smaller vesicles. Following that, there's a step for immunocapturing onto beads. So for that, the beads were first sort of incubated with the antibody. And before that step could be done, if you remember, we had discussed the fact that you want to build like a strong structure, a strong magnet, which can basically attract all the all the desired EVs or all the desired exosomes from the entire pool of exosomes. And to create that strong support or that strong scaffold, an initial step is required whereby the antibody of interest should be incubated with biotin. And then after biotinylated antibody has been generated, it is then immobilized onto a strong support, which in this case was a streptavidin bead, which is a heavy bead that during the centrifugation step would pull down all the immunocomplexes and they can then be segregated out quite easily. So after the functionalization step has been done, whereby you basically you immobilize or you localize the biotinylated antibody onto the streptavidin bead. Following that, they are incubated with exosomes, and then following overnight incubation, whereby you also subject it to rotation at 4 degrees centigrade, you are basically immunocapturing the desired exosomes subpopulation out of the entire pool. And following this, the immunocaptured exosomes were supposed to be eluted, and elution in this case was suggested using glycine HCl, which is acidic glycine at pH 2.7. And then after subsequent steps, finally, you use a concentrated, and what you end up with is your your purified exosomes of interest carrying the desired cell surface marker, which has been made possible because of the immunoaffinity based separation or isolation step. Following this, we looked at, so this completed our domain for non-invasive diagnostic or advanced techniques of cancer diagnosis, following which we moved on to conventional routes of cancer therapy. So basically this we went into all of the known techniques whereby it involves surgery, radiotherapy, and finally chemotherapy. So these are all the different types of surgical procedures that we discussed, like curative, preventive, and diagnostic surgery. I'm not going into details of it because they are just definitions. You can look at them in your own due time. And following that, we looked at staging, debulking, palliative, supportive, and restorative surgery, depending upon what purpose that surgical procedure basically serves. Uh, then we looked at the definition of chemotherapy, what is its goal, and how the principle behind the entire strategy. So basically, its job is to stop cell proliferation and to inhibit tumors from growing in size and to obviously limit their ability to invade and metastasize into other tissues. So we understood that, and we saw that at different stages of the cell cycle, different types of chemotherapeutic inhibitors or chemotherapeutic strategies can actually be used. So, like you can see, G1, S, G2, and M, each stage had different types of drugs or different types of chemicals that were more effective, and each of them have different jobs to do. So, again, you can go through that in your own due time, but the overall purpose was to disturb the cell cycle or either arrest the cell cycle or stop cells from further proliferating due to that arrest or calling for other machineries to come and take over and just basically compromise the ability of cells to divide. And as you can understand, the main challenge that was associated with this type of treatment was the fact that it was not very specific in nature, and therefore all the healthy cells within the body were also not equally, but were also quite susceptible to this sort of treatment, and therefore the off-target effects are quite huge, and that remains a challenge with chemotherapy till date. And we looked at examples of the nails or the brows and hair. All of this gets lost because these are areas of cell. Normally, under physiological conditions, these are areas of rapid cell division, and also in a lot of cases, the patients are also quite significantly immunocompromised. So that also remains an added challenge to the already ongoing treatment. So overall, this was the idea, and we looked at the different mechanisms which can be followed by different classes of drugs. And I had stressed on this the last day as well, and I would again iterate the same. You should be very clear about the mechanism by which some of these drugs work if you are looking forward to working in cancer biology, even if your focus is not really on um, drugs and on cancer therapies, it's something you can definitely be asked about. So it's better to be in the know. So we looked at some of the drugs which have a multi-pronged approach of basically affecting cancer cells. Like vincristine affected microtubules, and it also affected the mitochondria as well, where it disrupted the respiratory chain within the mitochondria necessary for ATP synthesis. So yeah, examples like these, it's better to be familiar with those. And finally, we looked at detailed mechanisms of these three drugs, which are cisplatin, pifluorouracil, and docetaxel or paclitaxel. And we looked at the ultimate result being the fact that cells will die, they'll be subject to apoptosis. But the way in which these cells are being pushed to the condition where they are eventually succumbing to cell death, that is quite variable. So these are all of the different mechanisms that you need to keep in mind. So this was our discussion for the last class, and we ended with these key takeaways and key questions. Following this, we'll be moving into today's class. If there are any doubts, you can let me know. And again, to iterate, we have 4 classes left. Today is session 8. So 9, 10, 11, and 12 are left. We've kept session 12 for the discussion sort of session. So I hope more people will be joining in. If not, I'll just go over and discuss the topics, like I've already said, the questions from each of the topics, and I'll summarize the entire course as well. So that'll be the plan. And in the upcoming sessions, we'll be discussing these few leftover topics. I've very briefly introduced precision medicine today. I want to go into a little more detail about that as well. And I had mentioned in the previous class that I want to discuss some models for studying cancer also very briefly. So we'll first wrap up these two topics, which are some understanding some key terminologies with reference to cancer. Some terms. These are like basic statistical terms. So I'm thinking we'll do this in the last class, second last class. And in the next class, I would like to move on towards drug resistance. And along with that, maybe a little more detail into precision medicine and also drug resistance at the same time. And what I would also sort of like to get you familiar with is the idea how people are now integrating artificial intelligence and machine learning algorithms into cancer diagnosis, cancer prognosis, basically in oncology, in the field of oncology, and how that is, it's definitely a something that needs a lot of work, but it is being actively done, and people are being given like a sort of framework as to how their entire prognostic life is going to look like, and what is their life expectancy, what is the probability that they're going to survive this, and what is the accuracy with which a person can be confirmed for sure that they actually suffer from the disease. So all of that's getting integrated. I'm also very new to this concept, but however much I understand, I'll certainly transfer the knowledge to you. And yeah, that'll be the plan for the next class. And in the following class, we'll look at these cancer statistic terms, and we'll look a little bit into details about the animal models. And following that, the discussion session, and with that, we'll wrap up the course. So that'll be the plan. People who have been joining in, thank you very much for being consistent. It helps me a lot. Otherwise, it's fine. I understand everybody has schedules, but thank you. Now, coming back to today's topics that need to be covered. First of all, we'll look at some current advances in cancer therapeutics. We'll look at the concept of drugging the undruggable. Before that, we'll understand why at all something is considered to be undruggable, what is the big deal. And thirdly, we'll briefly get some introduction into precision medicine. So with this introduction in mind, this is just sort of a historical perspective or a time lapse of the advances in cancer therapy. I had discussed something similar a while back, but this is it's not specific to any one type of therapy. It's just an overall perspective. So again, keep in mind that these are just some examples. It's one review paper that has okay, and just for your information, this review paper is pretty much what I have taken up for discussion today. It is a pretty comprehensive review. You can refer to anything which you find interesting, but this one I found very relevant to our discussion today. So most of the schematics you'll see are from here. So I just wanted to let you know that because I've not given the citation in all the upcoming slides. So first of all, let's look at very briefly look at the strategies to get some, to tie up our discussion from the previous class. Uh, so in the early 90s, or even before that, cancer treatment majorly involved X-rays and chemotherapy. Then following that, you can see that BCG, which is something you must be familiar with. It's a vaccine which is used. So that was approved for intravesicular vesicular installation for superficial bladder cancer. Okay, yeah, this is also something. It's not really a separate topic, so I haven't really put it as part of the course outline. But this is a concept you should be familiar with. That one thing which is very extensively explored in not just the field of cancer biology, but just in medicine in general, is the idea of drug repurposing. What does it mean? It means that whenever you don't have a very targeted treatment strategy for any condition, then what is the first objective? Instead of trying to design something new, instead of trying to figure out, say for example, let's take the example of COVID. So when COVID came in and nobody had any treatment in mind for that particular disease, at that point of time, the first approach was not to go for designing a new treatment, a new vaccine for tackling that virus. It was something that was done in parallel, but then the first objective was to try and repurpose something. So if you remember, at that time people were trying to repurpose anti-malaria drugs or other strategies just to, bring people in just to get people more, just for people to get more access to the existing drugs and to help in recovery of the disease a lot faster. So drug repurposing is something which is extensively done, and it's obviously a very useful strategy as well. So this was one such example, and it was done in as early as 1990. Following that, you can see there was the approval of the first cytotoxic monoclonal antibody. We will be discussing that today, for a treatment of B-cell lymphomas, then introduction of trastuzumab for breast cancer targeting the HER2 protein. This is also something I had discussed very early on, somewhere in the third or fourth session when we were discussing cancer cell signaling, and we had talked about receptor tyrosine kinases. So if you remember, there I had talked about trastuzumab, and we had seen how some drugs or some treatment strategies, they show some promiscuity, because of which they are able to, the same drug can be used for targeting different pathways, and effectively it leads to the same conclusion. Then came FDA approval for imatinib, the first kinase inhibitor. Alotinib, Brigvir, bevacizumab was the first anti-angiogenic drug. So anti-angiogenic, obviously, as you can understand, because angiogenesis is naturally a very key, very important hallmark of cancer. So this anti-angiogenic drug was a huge revolution in that regard. Then sorafenib, again, something you definitely must be familiar with by now. If not, just go back and look at your slides from the third or fourth session. I can't exactly recall. It was the fourth session when we were discussing cancer cell signaling and the way of targeting these signaling methods, and you'll find that there. Then around around 2011, the concept of CAR T cell came forward. This also we'll be discussing briefly today for leukemia treatment. Then in 2014, you can see a couple of things have been clubbed together. Like for example, BRAF mutant melanoma. For this, trametinib and dabrafenib were given approval. FDA approval of pembrolizumab was made, and nivolumab for melanoma treatment. This is something you again probably are familiar with, Gardasil. So Gardasil is a sort of a precautionary or preventive vaccine, which has been approved as a way of sort of not even bypassing, but it's again a preventive strategy against cervical cancer, which has been approved for women under 30 years of age. So Gardasil is something you would already be familiar with. If you want to, I think I would let you try and figure out how it exactly works. But it's a well approved, very easily accessible vaccine, which is recommended. So if you know women under the age of 30, obviously I think the recommended age is around 22 to 30. So just give it a little bit of a Google search, and it is very strongly advised that women should take this vaccine. It really lowers down the chances of cervical cancer in the long run, because as you can know, this, as you might know, is often the result of a viral infection. It doesn't always have to be genetic. So any woman can be prone to it. So having a preventive vaccine is definitely going to be helpful. So yeah, just for your common knowledge and for your practical application, if possible. Uh, then there was an approval of another treatment strategy for melanoma. Sotorasib, the small molecule inhibitor targeting KRAS gene mutation, I'll be talking about that as well. And there are some other antibodies, monoclonal antibodies as well, which were recommended for some patients. Again, if in case you don't remember the name of the drug, if it carries MAB at the end, it stands for monoclonal antibody. So it's an immunotherapeutic strategy for treatment of whichever cancer it is recommended for. So this was just a historical timeline based summary of all the discoveries that have been made uh, in context of cancer treatment. Okay, now coming to this slide, don't get overwhelmed. There's a lot of information. We'll go over it one by one. So the first discussion we have on the table for today is about small molecule inhibitors. So what do you understand by that? Very naturally, it is a the key distinguishing factor of these class of drugs is the fact that they're very small in size, which definitely would confer them some advantage because of which this entire class of drugs has at all been taken into consideration, right? Also, what you need to understand, it is an inhibitor. Okay, again, one more thing that like as you are learning, you should learn to distinguish the key differences between these terms. Like for example, if you're talking about a monoclonal antibody, like those drugs named as MAB, or if you're talking about an inhibitor, or if you're talking about a degrader, they are all very different types or different subclasses of cancer associated drugs. An inhibitor is something which is just going to stop a process. It is going to bind to some protein or some molecule within the cell, and it is going to stop a process or halt a process at any particular stage. A degrader is a different class of molecules. I'll be getting into that later on. A monoclonal antibody is going to take a whole other approach. It is also going to bind to a certain molecule, but what it does downstream of that binding is going to be quite different. So these, like, don't use them interchangeably, and understand that if you are coming across any article where each of these terminologies have been used, it means something very different, and they need to be used very carefully. Okay, so first look at the yellow box to understand what exactly these SMIs are. So small molecule inhibitors, they are a class of drugs which can interfere with or block the activity of some specific molecules by interacting with them. These molecules, they're typically proteins, and they usually play very important roles in cell signaling, gene expression, and metabolism. By binding to the target molecules, SMIs are able to disrupt their normal function, thereby interfering with disease progression, or they can treat certain conditions. Okay, so this is the basic idea. They'll bind to something, and then they'll uh, help in, they'll basically interrupt a certain process. Now, why am I saying interrupt? It's because sometimes some small molecule inhibitors can either completely shut down a process, sometimes they can help in aiding some other process. So there are different ways in which they can work, but majorly the class that we look at, because we're talking about cancer in general, the idea is that they are going to probably inhibit something, right? So let's look at this schematic. You might already be familiar with a lot of them here. Uh, something intro, just a second. Okay, I don't know. Yeah, so what you see here is a schematic which shows you the basic key pathways, key signaling pathways, which are necessary for a cancer cell. The first one you're already familiar with is the RTK signaling pathway, or the receptor tyrosine kinase signaling pathway. Also, keep in mind you're seeing two different cell types here. What is shown in pink is an endothelial cell which lines your blood vessels most commonly. And there you can see at the surface there is a receptor tyrosine kinase to which ligands are binding. And you can see that different examples of RTKs are given here, like VEGFR, PDGFR, FGFR, MET, etc. And this red, this symbol indicates inhibition. It means stopping a particular process. So either it can be an inhibition of the endothelial cell receptor, or the same thing is possible for the receptor which is present on the surface of your cancer cell or of the host cell, tissue cell. Let's put it that way. What I would like you to note is that the types of RTKs which are present on each of these cell types, they're quite different. If you look at the blue colored cell surface, which is indicative of a cancer cell. You can see ABL, KIT, HER, ALK, ROS, TRK, FLT3, all of these different types, which are placed in addition to all of the ones which are recommended for the endothelial cells. So that indicates that depending on the cell type, the type of RTKs which can be present, or in other words, the type of cell surface marker or cell surface protein that can be present, that can vary, and that is a property which is extensively explored for a large number of molecular biology techniques. But yeah, I just wanted to highlight that for you. So here as well, the cell surface receptors which are present in this cancer cell that can also get inhibited, or that is also a target that is used for inhibition by SMIs or small molecule inhibitors. Downstream of it, these are already pathways we are familiar with: the RAS, RAF, MEK, ERK, MAP kinase signaling cascade, or the mTOR based signaling cascade, or the JAK-STAT signaling cascade, or the focal adhesion kinase cascade. You can see that in each of these cases, you can have inhibition at many different steps. And the type of inhibitor in this case, like for example, in some cases, some inhibitors, because they have promiscuity, they might inhibit more than one factor. Like for example, something can inhibit, say, RAS as well as MEK, or RAF as well as MEK. So different types of inhibitors can work in different manners. The net result which comes out of all of it is that it is going to stop cell proliferation, or it is going to disrupt cell proliferation in the long run. Secondly, if you look at this entire cycle shown at the bottom, it shows you the physiological cell cycle process. And you can see that there are different steps at which inhibition can occur. Majorly, the focus is on CDK4/6 because it can be inhibited by small molecule inhibitors. And CDK4/6, I think an article was published day before yesterday, most probably, where some CDK4/6 targeting drug was um, cleared for clinical trials, something like that. I'll go to it. But basically, what I want you to understand is that these are areas of very, very, very active research. This review is from 2024. So usually reviews, they try to keep their focus on discoveries made in the last 4 to 5 years, because beyond that, the information is already too old and a lot of progress has happened. But these are still areas of very active research and getting published, as you can understand right now. So there's always more to learn. So what it effectively does is that it stops this transition from the G1 to S phase. It disrupts the G1 phase of the cell cycle by inhibiting CDK4 and 6. Following this, you can also see inhibition at other levels. Like for example, here you can see there's inhibition of cell surface receptors by hedgehog signaling. You can see inhibition of BTK. I have not given you the names here because I think it's just going to add to your confusion. You'll find full lists of all of these drugs anywhere. Or for that matter, if you just go to this article that I've mentioned here, if it is interesting to you, there's an entire table with all the FDA approved drugs for all of these different FDA approved small molecule inhibitors, which have been cleared for clinical use in the recent past. So you can look at that. Sorry. Also, you can see that there's CXCR4 inhibition that can also occur, and all of them are eventually merging in this particular location, whereby gene transcription or gene expression is affected. That too at multiple levels, like HDAC inhibitors, DNMTs, PARP inhibitors. All of this can also occur. I will I've not put it in today's class. I missed it completely. Maybe I'll just explain it briefly today if we wrap up the class early, or I'll come to it in the next class. There's an entire section of epigenetic drugs. Um, Again, keep in mind that epigenetic drug literally means something which is working above the gene, right? So these are drugs which are disturbing the way in which the chromatin is maintained and the way in which the chromatin is expressed within the cell. So they are working above genetics, and therefore, what you need to understand is that these drugs are not going to be permanent. Their consequence is quite transient, and therefore, epigenetic drugs are leaving a lot of open-ended. Like there's a lot of questions that are associated with their use. Having said that, these are drugs that are found to be so efficient for so many cancers because, as you can understand, that irrespective of what type of cancer it is, gene expression will have to be controlled, will have to go haphazard in whatever type of cancer it is. Say, for example, a patient with liver cancer, a patient with breast cancer. Yes, the way in which you would approach treatment of that patient initially, that would be quite different. For a breast cancer patient, you might go for hormonal therapy because that is more relevant to the type of cancer it is. For a liver cancer patient, you'll go for some other drug which is more relevant in that case. However, eventually, what you have to keep in mind is that if there is a somatic mutation that has helped in formation of that cancer, that mutation will have to get transcribed and translated, and that mutant protein will have to be synthesized. To facilitate that, transcriptional machinery has to work and has to work a lot and has to work irrespective of what errors might be present in the gene. So, therefore, disturbing the transcriptional circuitry is a brilliant challenge that people have taken over. So, the class of epigenetic drugs, they actually affect this transcriptional circuitry and they do it in multiple ways. Like, for example, I'll not go into more detail about it right now, but for example, DNMTs and HDACs, if you have studied a little bit about genetics, you would know this, that DNMTs are DNA methyltransferases, HDACs are histone deacetylases. What they basically do is either they transfer a methyl group onto the DNA, or HDACs mean they are deacetylases, which means they are removing the acetyl group from the histone, which is associated with the DNA. Effectively, what it does is it disrupts or it affects the normal transcription of that particular area of the genome. So using an inhibitor which does that, which interrupts DNMT action or which interrupts HDAC interaction, it will basically affect the transcription of that particular gene. Again, keep in mind that because it is an epigenetic drug, you can never completely ensure that only that gene's transcription will be affected. No, it is going to affect any and every gene which is very actively transcribed within the cell. As a result of that, there's a huge non-specificity which is associated with epigenetic drugs. So it's sort of a balance between the pros and the cons of using these drugs. So they have the limitations, but their utility and their potential is so huge that sometimes their shortcomings are overlooked. So this is an area of active debate, and I'll give you some examples. I don't have slides on that right now, but maybe we can take it up in the next class, or maybe I'll just show you using a simple search. But yeah, basically epigenetic drugs are a very interesting class that is still being studied, and that has its own fair share of questions associated with it as well. So these are all of the different types of drugs, and also effectively what it does is you want cells to undergo apoptosis. So any anti-apoptotic protein, if it is being expressed at very high levels, that is also a potential for therapeutic targeting and is used sometimes for patients as well. So yeah, this is the entire idea of small molecule inhibitors. What you see here are the mechanism or the mechanistic insight into the way in which SMIs work. So antibody therapies and small molecule inhibitors are the two main methods for targeted cancer treatment. The mechanism of SMIs is to inhibit the target protein's function by binding to the pocket which is present on their surface. SMIs can bind a wider range of extracellular and intracellular targets compared with antibodies due to their smaller size. Remember what I said, that the name small indicates that being small in size must definitely confer some advantage to this class of drugs, and therefore they have more potential, and therefore they're more interesting targets for study. So what is the main advantage associated with them? It is because they are smaller as compared to antibodies. They have a much wider scope of binding to different proteins. Say, for example, if a protein is very intricately folded, so it has a lot of smaller folds. An antibody, because of the way it is structured, if you've studied antibody structure, you would know that it has a lot of disulfide bonds, which give it that unique rigid structure. Now, that rigid structure often poses steric hindrance and it cannot always access inner pockets within proteins. And it may so happen that for a particular protein, that access is necessary so that this inhibitor is able to inhibit the protein's inherent function. In case this is not able to do so, therefore, in case the antibody is not able to do so, having a small molecule inhibitor for the same, because it's a, as the name suggests, a small molecule, it will be able to access that site and it will be much more efficient and it will be able to do the job a lot better. So that will be a huge advantage. Furthermore, because most small molecule inhibitors can be consumed orally, antibodies are at a disadvantage in that case as well, because antibodies mostly have to be injected, they have to be subcutaneously administered or intravenously administered. However, because this is just a chemical moiety, you can just consume it. So the translatability from bench to bedside is a lot more, and it can more easily be implemented for patients who are capable of taking care of themselves. Also, one major advantage is that I think I had introduced the blood-brain barrier in the last class. So this is actually one major advantage of small molecule inhibitors as well, is the fact that they are able to breach the or penetrate through the blood-brain barrier, and they can control intracranial lesions as well. So intracranial lesions as in any sort of mass which may be formed within the cranium, within the brain, or within the head, basically, that can often be accessed by small molecule inhibitors or um, any entity which is very small in size. On the other hand, antibodies are not able to do so, A, because of their size, and B, because these are areas which are often considered to be immunoprotected. They are deprived of any sort of immune access, and therefore using an alternative strategy is actually quite smart and quite useful, and it is being done quite actively. Uh, Before going to this example, if there are any questions, let me know and then I'll move ahead.
No, ma'am.
Okay. Okay, so we'll move ahead. Um, Now the theme for this slide is drugging the undruggable. So why at all are we really so bothered about this thing? Um, I think it is there in this slide. Okay, this has shifted somehow. Anyway, you should be able to understand, I think. Yeah, these have shifted. Basically, this subheading for the slide was a covalent modification or covalent modulation of small molecule inhibitors. The question is, why do you need that? This is because one major challenge that is faced in cancer is the fact that its most promising one of its most promising targets is the transcription factor, which regulates all of these different pathways, like I just explained. So, while it is a very promising target, one major challenge remains the fact that it's very difficult to target these proteins. Why? A, because they're usually individually, these proteins are not very huge in size. And apart from not being very huge, they do not always have a very clear binding pocket. So, by a binding pocket, what do I mean? Like, think of a protein. If you have a protein which looks like a sheet, it is going to be very difficult for a drug, for any inhibitor, for any molecule to come and bind to it. Versus if there's a crevice, if a molecule binds, it is going to get stuck, and therefore the efficacy of such a drug would be much higher. So it will, the affinity would remain, or the affinity or the tendency of that particular inhibitor to bind and stay, remain in that bound condition would be a lot higher. So this major problem is faced with any transcription factor in general, unless it has a very distinctive structure. And therefore, despite their huge potential, they have not emerged as very good, practically usable targets for treatment of cancer patients. One such example is the KRAS protein. So if you remember, we had talked about RAS, RAF, MEK, and ERK, and among them, the different type of RAS proteins as well. We had discussed in the third, or I think the fourth session probably. So we had discussed all of this. You'll get information about it from this slide. So KRAS has been found to be a very important target, and particularly a one single mutation which is found to be very, very abundant in a lot of patients is the G12C, which is G12 to C, which means from glycine to cysteine, there is a uh, missense mutation which has occurred, and this is found to be present in approximately 80% of non-small cell lung cancer. So imagine how abundant it is and how important this particular mutation must be. So out of 10 patients, if you can save 8 patients, that's a huge statistic. First of all, it's not something you find very easily. Um, Secondly, as promising as it is, challenge remain the fact that KRAS protein, if you look at it just overall, this is just a space filling representation of or the model of how the protein structure actually looks. So if you look at it from the perspective of a structural biologist, you would understand that this structure is considered to be almost smooth, and therefore it was considered undruggable. So this had remained a challenge for a very long time. If you just go back to the paper where people have first figured out that KRAS is a major, troublemaker in context of NSCLC or non-small cell lung cancer, from that time till as recent as 2021, this is how long it has taken for targeting a protein as promising and as important as this. So let's read about it a little bit more. So this particular small molecule inhibitor, which is sotorasib, otherwise known as AMG 510, was approved by the FDA in 2021. It is the first small molecule inhibitor that targets some specific KRAS gene mutations. It leverages the unique chemical property of the cysteine residue, which is present in this KRAS G12C mutant, to covalently bind to only this residue. What advantage does it give you? Very the most obvious answer is that the fact that it only is able to bind to the mutant protein and not the normal one gives it a huge advantage. What are you doing? You're cutting down the chances of any any non-specific or any off-target effects to a huge extent, because you are only ensuring that only if the mutant is present, only then will my drug actually bind. What does that do? Because it has covalently bound to this residue, it locks KRAS G12C in an inactive state. As a result, it is not able to bind to GTP, and therefore all the downstream signaling pathways would be shut down. So just for reference, look at this slide. He's making noise. But yeah, basically look at this step. This is where KRAS is going to work. And if at this step, if at this step you are ensuring that it is not able to bind to GTP, it is not going to get activated. And as a result, RAF, MEK, or can all the downstream pathways are going to be completely shut down. So as you can understand, this discovery was immense, huge, and a huge, huge revolutionary contribution to the field of cancer therapeutics. This launch of AMG 510 defined a milestone or signified a milestone in the undruggable proteins history. So you can see that this is present in, this is going to bind. So this particular drug, don't know if you're able to see, but yeah, this is sotorasib. It is able to go and bind to this specific mutation present within the KRAS protein, and it is able to covalently modify it and stop the entire process of cell proliferation, which can be controlled by KRAS right at that step. Okay, so this was one huge example. Yeah, this has sort of shifted a little bit when I converted it to PPT, but yeah, it should make sense to you either way. Basically, here we're looking at the second approach of targeting the so-called undruggable. So that is by allosteric inhibition. So if you have studied enzymology at some point of time, you would understand what allosteric basically means. So what these inhibitors do, they actually bind to other sites of the protein. So um, you have a particular any protein, any enzyme or any functional protein. If you have that, there will be one binding site which is called as the ligand binding site, which is the key location where your protein of interest main target or cognate target is going to bind. On the same protein, there is some other site which is considered to be the allosteric site. And what happens is that the allosteric site controls the conformation or controls the accessibility of the main ligand binding site. Say, take this protein, for example, if this is your protein of interest, here is where the ligand, suppose, for example, binds. At this location, you have another site, which is the allosteric site. So when something comes and binds to the allosteric site and somehow inhibits it, it may happen that this causes a conformational change, as a result of which this initial binding pocket is no longer accessible. So the protein's um, normal physiological or biological function is going to be going to be severely compromised. One such example of this is the BCR-ABL inhibitor. So I think I had discussed BCR-ABL when we talk about chromosomal fusions, gene fusions, etc., as some examples of added, you know, I think, yeah, when we talked about tempress to ERK fusion, BCR-ABL fusion, all of these are chromosomal anomalies which are found to be very prevalent in certain types of cancers, especially the BCR-ABL one is found to be prevalent in context of leukemia. So, what exactly happens there? Here, what you see in on the left is the active protein, which is the active BCR-ABL protein, and this is found to be a huge contributor to the development and the progressive um, harm that it causes, leading to chronic myelogenous leukemia. What you see with it is the addition of asciminib, which is ABL001, which is an allosteric inhibitor for often used for CML patients. And what does it do? It basically locks the BCR-ABL1 fusion protein in a very inactive state by binding to the myristoyl pocket known as STAMP. What does it help in? It helps overcome drug-resistant mutations such as the T3 315I mutation, which is threonine to isoleucine mutation. So this is one example where the BCR-ABL normal protein is inhibited by small molecule inhibitors, which bind to the allosteric site and inhibit the function of the fusion nonsensical protein, and therefore they are used as potent therapeutic agents in context of certain cancers, in this context leukemia. Another example is cobimetinib, which is also known as GDC0973 or RG7420. It has been recognized as an oral MEK1 inhibitor for melanoma research. And as you can see from, it's also a RAS inhibitor basically. So the MEK1 is one of the MAP kinase enzymes. So it also disrupts this entire pathway. So you can see that RTK, GRAP2 sauce, RAS, RAF, and then from RAF you have MEK, or you have MEK via MEK, you have ERK, and from ERK you have nuclear substrates which are getting phosphorylated, or cytosolic substrate which are getting phosphorylated. So using an inhibitor, um, just a second. Yeah. Using an inhibitor for MEK1, which is working in this cascade or is working in inhibiting this particular cascade, is going to shut down this process, and it has been extensively studied in context of melanoma. These were just two examples, like asciminib and cobimetinib. IB is usually indicative of some sort of an inhibitor. I'm just explaining the like the terminal ends of all of these enzymes names for your better understanding. So MAB was for monoclonal antibody, and IB is for inhibitors. So that basically sums up the ideas of small molecule inhibitors that I wanted to discuss with you. Now coming to the next class of drugs. So if you remember, we had talked about the undruggability. When we were talking about the undruggability, we talked about different types or different subclasses of drugs, and among that, I had said degraders, I said inhibitors. Issue with it. It's lagging. Anyway, let me check. Okay, fine. So what we are discussing in this slide actually belongs to the group of degraders, which is the third subtype that I had mentioned previously. For that, what you need to understand first is the concept of PROTACs. What are PROTACs? PROTAC stands for proteolysis, targeting, chimera. Now, if you understand chimera is basically a fusion product. You would understand it if you have already studied a little bit about recombinant DNA technology, and you would understand that chimera is basically it does not have one unique moiety, but it is a combination or a the combination product of more than one type of thing. So PROTACs, in PROTACs, PRO stands for proteolysis, TA stands for targeting, and C stands for chimera. What does it exactly mean? It means you are generating a chimeric molecule which triggers proteolysis, which means protein mediated degradation of a certain molecule. PROTACs are a strategy that utilizes protein degradation mechanisms to eliminate target proteins by simultaneously connecting the target protein with an E3 ubiquitin ligase to form a ternary complex molecule for the degradation of the target protein. What does it mean? One by one. First of all, you have uh, you have three sort of entities which are all fused together. The first one here, you can see it is shown in green, is the ligand for your protein of interest. Take an example, say if you are looking to target, what do we take? Say you have AR, androgen receptor, that you want to target. Then you would have to design the proteolysis targeting chimera or the PROTAC in such a way that it has one molecule which acts on one part of the molecule which acts as a bait. Bait basically means you are, you know, trying to attract something which is going to eventually get sacrificed. You've all understood the concept of baits at some point of time before, but basically that is what it does, and this is called your bait moiety. What you see in pink is your it sort of helps in degrading. So this is your E3 ubiquitin ligase. It is a ligand for your E3 ubiquitin ligase, via which it will go and fuse or it will bind to the machinery which helps in proteasomal degradation. And both of these are linked by something you can see in gray. It is a linker, which is by itself not going to cause any changes to other conditions. And what this entire molecule altogether is known as is a PROTAC. So, therefore, that's what the definition also wanted to explain basically. So, it eliminates target proteins by simultaneously connecting this target protein with an E3 ubiquitin ligase, and that forms a ternary complex molecule which is going to target protein degradation. So here is your ternary complex, whereby you have the ligand or the linker binding to the protein of interest, and you have the E3 ubiquitin ligase binding to the E3 ubiquitin ligase ligand. Following this, what happens is that because it is an E3 ubiquitin ligase, again, this is something you would need to have a little bit of background on. I hope you're all familiar with the basic strategy, um, which is used for ligation, and eventually it is going to lead to degradation of a protein. So there are um, how do I explain it? Basically, you have understood the protease. If you've understood the proteasomal degradation machinery, you would already know this. So our within our cells, for maintaining homeostasis, or depending upon whatever is the external condition, a lot of times cells have to actively choose to degrade certain proteins, A to release energy, B to give us resources, C because it is not required, and keeping that protein stable is actually requiring more energy than is worth. So under those conditions, to ensure protein turnover, this machinery comes into play, which is known as the degradosome, which is the degradation machinery. Within that, what happens is that there are certain sort of, think of them as degradation marks or degradation signals, which are installed onto your protein of interest, which needs to be degraded. And that calls for E1, E2, and E3 ubiquitin ligases. What do? They help in the recruitment or the attachment of this purple colored molecule known as ubiquitin. And when there is tandem addition, it leads to the formation of a chain known as polyubiquitin. And then that becomes a signal for this particular molecule to move towards the proteasome, where it can be chewed down and completely degraded. So the development of anti-cancer PROTACs primarily uses E3 ligands, E3 ligase ligands such as CRBN, VHL, MDM2, IAPs, AHR, etc. Currently, PROTACs have been developed for various targets to combat solid tumors and malignant hematological cancers, such as those which target AR, those which target ER, and BTK. These drugs have entered clinical trial stages with bavdegalutamide, ARV-471, etc., showing their therapeutic effectiveness. The PROTAC targeting MAP4 kinase 1 has the potential to be the first in line therapy, mimicking the PD-1, PD-L1 targeted therapy, while PROTACs targeting BCL-XL and ALK have shown broad spectrum antitumor activity, effectively killing leukemia and solid tumor cells both in vitro and in vivo preliminary preclinical experiments. So basically, there's just some examples given. For example, for those for AR, ER, BTK, etc., and some of them have actually been are suggested for being translated to clinics, but then uh, a lot of checkpoints are in place before that actually can be facilitated. We'll move on to antibody drug conjugates soon, but any doubts anybody has from PROTACs and from what we understood about allosteric inhibition, please let me know.
Why it is called undruggable?
Mhm.
Why is it called undruggable?
They were considered undruggable, like I explained initially. So the default condition is that for any protein to be drugged, say, I'll give you an example. Practically, how is it done? Say, for example, you are a part of a lab where you have like panels of small molecule inhibitors. So that's actually done in a lot of labs which are um, sort of multidisciplinary in nature, and you have chemists as well as some biologists working in the same lab. So what they do is they generate certain molecules which are, how do I explain, which are effectively going to kill cancer cells, right? They generate molecules like that. What is their objective? They want to find a target for it. So what is done is, in collaboration with biologists, what you do is you screen. If somebody, say for example, I as a biologist have one protein of interest, which I think is a great target, but I don't have an existing inhibitor for it, then what would I like to do? I would want to know if any other lab, any other researcher has any available chemicals which can inhibit the same. So what I can do is I can give them my compound and they will screen for their existing molecules and they'll see if anything is able to bind, and then they'll suggest that, okay, these XYZ compounds are very promising. Now, this is the most ideal situation. In which case does it work? It works if the protein has a very it has a structure which has a lot of crevices or a lot of folds, and this is majorly the case for proteins which are this is not the case. This is the major challenge with proteins which are just transcription factors in nature. They usually do not have a lot of crevices. That is because what is their job? Think of a transcription factor, just a transcription factor. Its job is to bind to DNA, and its job is to interact with all the transcriptional regulators. So I think I'll show you this. Remember, where is that image? Okay. Think of this. Say, for example, these two proteins, these two molecules which are forming the cell surface receptor. For them to be able to bind a ligand, their structure needs to have certain crevices where the ligand will bind properly, right? Take that in contrast with another protein which is just going to bind DNA. The maximum they might have is the crevice through which they're able to interact with DNA properly. That is the only possible location where you can target a transcription factor. Apart from that, they usually do not have any crevices, and that is why finding an inhibitor, a direct inhibitor for such sort of complexes is very difficult. Secondly, even if you can find some sort of a crevice, you might end up with a case where that molecule of interest not just binds to this particular transcription factor, but because that crevice is so generic in nature, the amino acids which are forming that crevice are so generic in nature that they can go and bind anywhere. In that case, you would have a lot of non-specific, lot of you know off target effects, and that is again not desirable. But the undruggability part comes from the fact that they are transcription factors and they're very difficult to directly inhibit. They are either present inside the nucleus, so having an inhibitor which can go through the cell membrane, go through the cytoplasm, pass the nuclear membrane, and then go into the nucleus, that is very difficult. Even if that molecule is able to go inside, it might not be very effective at stopping the transcription factor directly. So a lot of other different strategies are being taken into account. Small molecule inhibitors are just like an introduction that I'm giving you, but just Google it. You'll figure out how much research goes into targeting transcription factors because I'll tell you, I'll give you some insight into it. It's very interesting. I've not put it in this course because it's a fairly advanced concept. But basically, there are strategies which are how do I explain? Like they're antisense oligonucleotides. Basically, what you do is, let me just switch on the video for this. Think of a transcription factor X, right? This is how they think of this as the transcription factor. This is where it binds DNA. So what people have started to do is they have started to look at this region where it has the DNA binding domain, and they have started to try and inhibit that. Now, what is the challenge with it? If you think of the DNA binding domain, any protein that needs to bind DNA, it will have certain there are certain types of motifs which are required for a protein to have the DNA binding capacity. Like, for example, they have a zinc finger or they have a leucine zipper. These are basically some molecules which help in better interaction with the DNA. That is all. But for it to be targeted, for it to be inhibited and to disrupt the ability of the protein to actually bind to DNA, you need to have a very specific way of targeting that protein. What people have started to do is one very interesting way is they are making molecules, making peptides and making short nucleotides, like short oligos, basically double-stranded oligos, which look exactly like or which have the same similar residues like that of the original protein. Say, for example, if I have any protein, say ERK or MYC, these are very commonly, very potent, very common transcriptional factors which are present in many types of cancers. Now, the region where MYC binds on DNA, if you just take that sequence, that DNA binding region, and you design that as a drug, you modify it in such a way that it will have very high affinity for your protein of interest. Then what will happen? Within the cell, within the cytoplasm, or within not even the cytoplasm, let's say within the nucleus itself, you will have a lot of protein which is free, and you will have some protein which is bound to DNA. When you are giving a surplus of this particular drug, which is acting as a inhibitor of that DNA binding region, then it will go and bind to that protein, and it will prevent that transcription factor from binding to DNA. So while it sounds theoretically quite smart that, okay, you are just giving the protein some sequence to bind on, as a result of that, it is not exactly able to do its job. It sounds like a very smart and very foolproof strategy. But imagine the challenge that comes with delivering such a sequence into the cell. You can't just put DNA into a cell. It will not take it up that easily, right? You are not performing bacterial transformation. You are delivering an oligonucleotide sequence into a mammalian cell, into a cancer cell, and it has to go through so many barriers. So there's a lot of challenge that comes in. And honestly, while it seems very promising, its translatability into clinics is it's a far cry. But yeah, these are very like people are trying to look at very bypass strategies to try and inhibit transcription factors because of that undruggability factor that I told you about. But yeah, using such covalently modified inhibitors for a particular mutation, that is quite useful. But again, see, it has its own set of challenges. Not every patient, say for example, that drug with non-small cell lung cancer, not every patient is going to have that same mutation, right? So in case you have to give that drug, you will have to screen that patient. You will have to first check whether this patient carries this mutation, and only then will this drug, using this drug, make sense. Also, just using this drug is not going to be enough because KRAS mutation is a driver mutation, but there are n number of other mutations that could also help and aggravate the oncogenicity of these cancer cells. So you have to give it in combination with something else, some other generic cell proliferation inhibitor or cell division inhibitor. So there are a lot of challenges with targeting transcription factors. It's the delivery is a huge challenge. Like people have tried encasing it within liposomes. They have tried coating it so that you basically fool the cell into taking it up. They have tried tagging it with different sort of things so that uptake is easier. A lot goes into it. So yeah, this definitely remains a challenge. And if someday we are able to figure it out, it's going to be a wonderful thing. And in fact, what I just explained about Protax now, this this concept of using a bait moiety to uh, sort of attract your protein of interest, this has been sort of modified, and a concept known as traftax has been brought into. It's still a concept that is getting developed. It's not a very foolproof method, or oligoprotax as it is more commonly known. So there what you do is, instead of having this ligand as the bait, people are using that sequence, binding sequence, as the bait. And what that will do is, you will basically call, or you'll basically pull that protein of interest. So for example, if like we were talking about MYC, if you give the binding sequence of MYC here, then MYC is going to bind on top, and then it is going to get degraded and you will be able to save the cancer cell. But again, this is the most idealistic situation. That's not how cancer cells work. And it has a lot of challenges. So people are carrying out research. They're first checking things in cell lines. They're then going to patient into models which replicate the situation in patients. Then they're going to go through animal studies. It is going to go through clinical trials. So it's a long way to go. But yeah, this is overall the challenge with undruggability and the way in which undruggability can be tackled, or at least is being explored for tackling. Again, you never know where research is going to take you in the next couple of years. But yeah, these are areas which are being explored. And also, one more thing I would like you to read about. Um, I think I've put it in the key takeaways and question slide as well. Um, think about this as well. Say you are working on a certain cancer. Say you work on let's take renal cancer, for example, and you have figured out that, let's take lung cancer, easier example. You have figured out that a certain protein X has a certain mutation Y, and that is a major driver of cancer in a large number of patients. Now, what you do, you collaborate with another lab and you design a PROTAC. PROTAC, as you can understand, is something which can be designed very easily. So that lab gives you a PROTAC which has your ligand for protein X, and it is attached to a linker Y, and therefore you are able to degrade that protein. All good, looks very good. So, what would you think is the major challenge that would happen in that case? What is the major problem? First obvious issue that you might face with completely degrading a protein. Think about it. I'm not putting pressure on you to think about it right now. We'll discuss it in the upcoming slides. But there are very direct consequences of that as well. It is very similar to what you might see for an epigenetic drug. Basically, you are completely removing a protein from a system. A, is it going to be very foolproof? B, how long is the effect going to last? C, will the cell survive? Will a normal cell survive? D, how is it going to pass through all of the different barriers? So, just a couple of things to keep in mind. Just read about it a little bit, think about it a little bit, and of course, I will discuss it pretty soon. But yeah, these are very interesting areas, but they have really big challenges associated with them as well. So yeah, okay. Coming back here, I diverted a lot from the question, but yeah, these were just topics. I've not put it in the slide, so I thought I'll just at least get you familiarized with the keywords, and then you can then you can read about it in your own time. Or if it is interesting to you, just let me know. We'll discuss it later on. But yeah, this is what I wanted to introduce you in context of small molecule inhibitors and PROTACs. Now we'll move a little bit away from the strategies we have discussed and we'll look at antibody drug conjugates. So you remember we had talked about monoclonal antibodies very initially. So conceptually, just to understand something, initially the concept that was, in fact, a concept that is used even today of cytotoxic monoclonal antibodies is that you design an antibody. A monoclonal antibody is something which binds to only one type of epitope present on one particular type of antigen. Say, for example, if you have EGFR, let's take our example that has been consistent throughout all our classes. If you have a protein EGFR and that has, say, just a hypothetical number, say it has 500 epitopes on its surface. Epitopes as in 500 places against 500 regions on the protein against which antibodies can be synthesized. Then a mixture which has all those 500 types of antibodies will be called as a polyclonal mixture of antibodies, and these are polyclonal in nature. Each of these antibodies could also possibly bind to different regions. However, if you have a mixture, not a mixture, but a solution where you have purified one type of antibody which only binds to one epitope on one protein, then that is a monoclonal antibody. Okay, so the population of or a solution of all such um, or not a solution of lots of antibodies which are only binding to that one location, that will also be monoclonal in nature. So here, this is your antibody. It will bind to certain regions which are unique to particular protein. So the initial concept which was used was cytotoxic monoclonal antibodies. So cytotoxic basically means that if you use this, if you treat a particular patient with this antibody mixture or this immunoglobulin, what it is going to do is it's going to bind to those cells which have this particular antigen, and it is going to trigger cytotoxicity. How it does is you have to read about it. It's a concept of immunology. Basically, it's a process known as ADCC. So antibody dependent cell cytotoxicity is what it stands for. Read about it. I did not go into it here. You can, it's simple, it's just conceptually understand it. You have an antibody binding to a cell, and this cell is obviously cancerous in nature. That is why you have designed an antibody against it. So it recognizes this particular motif, and as a result it calls for all of these cytotoxic molecules present within the patient, and it causes that cell to die. Now this I'm explaining just between one cell and one antibody. Imagine this for an entire tumor and among different tumors present within a patient. So effectively, it is considered to be quite useful. And cytotoxic antibodies are still very much in practice. But yeah, their efficacy has actually been improved a little bit more using this concept of antibody drug conjugates. What does it what does it exactly entail? So ADCs or sorry, antibody drug conjugates, they are a unique class of drugs which are formed by conjugating these monoclonal antibodies which specifically target antigens with cytotoxic small molecule drugs. So what you're doing is you have this antibody, and onto its heavy chain, onto its constant region, you are localizing or you are chemically conjugating a certain type of toxin. So what it will do is, instead of affecting normal cell, this toxin is going to, you're basically um, refining the refining the delivery of the toxin. So instead of leaving it in the system, instead of a systemic way of removing cells from the body, what you're doing is you're targeting it only to the cancer cell. So this innovative system is often referred to as a biological missile, as it mimics the concept of a missile where the antibody serves as the guidance system and the toxin will act as the warhead. This allows for precise and targeted strikes on specific tissue targets within the human body. Once the ADC drug enters the bloodstream, the antibody component recognizes and it binds to the specific antigen which is present on the target cell. Subsequently, the ADC antigen complex is internalized by the target cell through endocytosis, and inside the cell, the toxin is released after being degraded by lysosome ultimately leading to apoptosis of the target cell. So we'll just look at it using the schematic to understand it a little bit better, but I think conceptually you must have understood it by now. So this is your antibody drug conjugate. What are some key points that have been explained here? What is the antibody being mentioned at? It should have low immunogenicity, fully human or humanized antibody. It should have very high specificity to your target of interest to reduce any off-target risks. Thirdly, formation of this antibody antigen complex should promote phagocytosis. What does this first statement mean? Why should it have low immunogenicity? It should have low immunogenicity because what you'll do is you will introduce a foreign antibody into a patient, right? So just the introduction of the antibody should not elicit an immune response that will lead to a non-specific immune response, and the patient's body will clear this antibody out of the system. So your drug delivery will be of no use. So in that case, what you need to ensure is that the antibody itself is not immunogenic, but once the antibody has recognized this particular antigen on your target cell, it is able to trigger the death of the target cell, and itself it should keep on circulating. So it should bind to more and more receptors present on more and more target cells, and it should be able to trigger their death as well. Apart from this property, the linker molecule helps in conjugating toxins to antibodies, and the toxin by itself should be a very potent cytotoxic compound, which is usually preferably of natural origin because that'll be a um, better drug than using something which is chemically synthesized because already you are using an external uh, source of delivering the drug to the cell. So once this antibody, this is just a schematic representation. This is not how an antibody binds. If you've studied immunology, you know already. But yeah, just for understanding's sake, you can see that the antibody has recognized its specific antigen on a target cell. And as a result of that, it is undergoing phagocytosis. Phagocytosis means it is getting engulfed into the cell. So here you saw this was one of the properties which was desirable for that antibody. After it has been phagocytosed, it fuses with the lysosome. Phagolysosome fuses, and then it triggers the toxin gets released, and then it triggers DNA damage or whatever effect is necessary. And simultaneously, it also calls for more and more. Um, it calls for more and more molecules which will come and degrade and cause apoptosis of that cell. So these are the different components of the antibody drug conjugate. First is your target antigen. What is its function? It is to ensure that the drug is very potent in nature. The target antigen should be very highly specific. It should allow endocytosis. Your antibody should be able to target that antigen. It should have very high affinity. It should be able to trigger ADCC, like I mentioned, antibody dependent cell cytotoxicity, which means that once this antibody has bound, it should be able to trigger cell death. It should have long half-life, which means that it should remain in circulation for a extended period of time so that you don't have to continuously inject a patient with very high doses of antibody. That's also not desirable. That's going to lead to more and more off-target effects. Sometimes it can also trigger autoimmunity. It's just going to make things work worse for a patient who is already suffering so much and is undergoing such strong treatment strategies as well. Uh, Low immunogenicity, cell permeability should be high. Linker, which means it links the antibody and the toxin, like we just saw here. It affects the release of toxins within cells. This linker should have high stability, specificity, high hydrophobicity, should be metabolizable. It should get metabolized within the cell. Then the toxin should be very efficient at killing the target cells. It should have very strong cytotoxicity, high structural remodeling capability, which means that when you form that antibody drug conjugate, it should be stable, but it should remodel itself to form a concise moiety, which is easy to deliver into a cell. Um, it should not be degradable. It should be non-degradable within cells. It should have strong hydrophobicity and membrane permeability to exert bystander effect. It should also have short half-life, which means that the toxin should not persist for very long in circulation. That might actually lead to more off-target effects. It might start killing normal cells as well eventually. Coupling method impacts drug uniformity, drug loading. So these are just different technological terms. I'm going to bypass it because I'm also not very familiar with it. But it's basically the way in which this antibody is generated, antibody drug conjugate is generated. So what it effectively does, just to sum up the whole thing, is you have your antibody of interest binding to your antigen of interest on your target cell of interest, releasing the toxin into the cell, allowing that to be taken up and degrading DNA or causing some sort of damage within the cell, which leads to cell death. Simultaneously, it should also be very good at calling for the cell cytotoxicity managing cells, like for example, the TC cells, which will effectively lead to more and more degradation and they will help in clearing out the tumor. So that is how antibody drug conjugates work. So I think we have just a few more topics left. So any doubts, you can let me know. If not, we'll discuss it at the end of the class, and I'll just wrap up the topics for now. Otherwise, I think we'll run out of time. So, yeah, I'll move ahead with this for now. I'm going to give you a very brief introduction on precision medicine. There's a lot that comes under this umbrella. Again, keep in mind, this is an umbrella term. Precision medicine, the definition I'll explain to you, but there's a lot that comes into it. I'll briefly discuss it. So, Institute defines precision medicine as follows. Precision medicine, generally considered quite analogous to personalized medicine or individualized medicine, is an innovative approach that uses information about an individual's genomic, environmental, and lifestyle information to guide decisions related to their medical management. The goal of precision medicine is to provide a more precise approach for the prevention of the disease, the diagnosis of the disease, and its treatment. So, summing it up, what does it mean? It means you use information about the patient to treat the patient, to diagnose the patient, and to prevent them from getting the disease in the future. That's the crux of the story. There are different tools of precision medicine. Like, for example, you use the social and behavioral data of patients. You use the omics data of the patient, whether it is their transcriptome data, which means their RNA information, their metabolome data, their genomic data, their epigenomic data, and their proteome data all together. And you use information from digital health and the information of the surrounding environment. All together, it is used to like all of this information about not just one individual, but about individuals within a certain population, within a certain country or a cohort, that all that information is collated and it is used for analysis and it is used as a platform for screening patients for different types of treatments. And it involves the use of all these technology like big data, AI algorithm, system bioinformatics. So you basically create a repository of information. You have a huge database of information about cancer patients or about patients in general. We are talking in context of cancer. So you have information about cancer patients from different countries. I think I explained it in one of the previous classes as well, where I discussed CBioPortal. So CBioPortal is one such platform for accessing all of these publicly available data sets. So you'll get information about different populations, and each of those data sets are published as if you go through those data sets, you'll see the TCGA Cancer Genome Atlas, which was published for this population in this year. You'll find some information about African population published in a certain year, about the Indian population you'll find in a certain year. So there are repositories of information about patients. The patient identity is obviously not revealed, but you'll have information about the disease. How does that help? That helps you in understanding how diverse the disease can be. So, the larger your sample size, if you're able to study 10 patients versus if you're able to study, say, 500,000 patients across the world, you will have a lot more information. You'll understand the disease a lot better. You'll be able to figure out which type of drug is more relevant for a certain population, whether one type of mutation drives the disease in one population, whether it is genetically heritable, whether it is something which is environmentally impacted, like I explained, Gardasil, for example, because it is often an environmental input which is coming in instead of something being genetically passed down in a lot of circumstances. So all this information, the more information you have, the more you can actually analyze the better you can design drugs, the better you can design ways of preventing and of diagnosing. So, how has this been a major, like, sort of a paradigm shift? That is because the traditional medicine, conventionally, what we have studied till now, is that you have the approach called as one size fits all. This like a phrase which is very commonly used for traditional medicine. So you would the idea used back in that day was that if you have a drug for a particular disease, all patients are treated with that drug. Somebody has lung cancer, they'll be treated with drug X. Somebody has prostate cancer, they'll be treated with drug Y. That approach has shifted from that perspective, and now people are looking more towards the idea of precision medicine. So it is taking into account subpopulation variability. In what kind of context? In context of their genetic differences, social differences, environmental and lifestyle differences, to propose therapies which are more specific, more precise to their disease. It aims for accurate measurement of the molecular, environmental, and behavioral factors which contribute to their normal health and to the disease, leading to a more precise diagnosis, leading to more rational disease prevention strategies, better treatment selection, and of course, development of newer therapies. So, this is the overall concept of precision medicine, which is the approach that we have shifted towards now. Now, the idea I told you I'll be discussing in one of the future classes will just go briefly into the use of AI/ML in this context, that how has it actually helped us in um, collating the information. So, think about it in this fact: somebody has given you um, say from in your books from class 1 to class 12 to study in one day. It's not going to be possible, right? So you have a lot of information, but the problem with that you'll face right now is actually interpreting that information, storing that information, keeping it accessible. So all of that has been facilitated by the integration of big data, by the integration of algorithms which help in uh, storing that information, recognizing patterns within that information, and using that as a screening tool to check that, okay, if a particular pattern has been recognized, then that pattern can be used for diagnosis, that can be used to say, for example, if from a lung cancer data set, if a particular algorithm is able to recognize that this combination of five mutations is diagnostic of lung cancer, then what it will do is the next time a patient is screened, it will check whether or not that particular pattern or that particular panel is present. If so, there is a very high possibility that the person possibly is going to develop lung cancer. There will be additional tests as well. There's a lot of information that has to be added to make it confirmatory in nature. But as a preliminary screening, it's doing a fabulous job, and it is very extensively being researched in today's time. And very soon you can expect it to be something which is almost going to be accessible to everybody. Anybody will be able to do that. And it's actually an integration for the better. So yeah, that is the idea of precision medicine. One example of precision medicine that I'm going to discuss today is the idea of CAR T cell therapy. Now, please keep in mind that CAR T cell doesn't always have to be considered to be an example of precision medicine. The reason I'm explaining that is because it's the initial idea of CAR T cells was to make it very personalized, make it very individual specific. So the patient's own CAR T cells were engineered or were generated, and that was used to treat that patient themselves. In the very recent past, there has been a lot of modification to generate a more off the shelf CAR T cells, which can be used for a larger subset. Because, as you can understand, if suppose, for example, if a particular treatment strategy has to be designed for me and only me, A, that is going to be very expensive. B, not everybody can afford it naturally. C, it will have very limited applicability for other patients, right? So the amount of resources that will go into helping one patient versus if that same amount of resource can be held for treating 20 patients, of course, people will try to go for the second option. So the idea being to look for the greater good, right? So in recent past, some modification has happened, which is why I'm telling you not to take this as a very hard and set example of precision medicine, but it's a concept which falls under that umbrella term. Okay, so let us understand this briefly. I've not put too much detail about it. This is more towards cancer immunotherapy, but we'll at least familiarize ourselves with the concept. So, CAR T cell, CAR stands for chimeric antigen receptor. So let's go through the schematic and then we'll read the legend. So how is CAR T cell therapy used to treat cancer? So first of all, you have a patient, and from the patient, their blood is collected so that you can obtain T cells or T lymphocytes, which is collected by health providers. What do providers do? They isolate or extract only the T cells, and the remaining blood is returned to the patient. From here, the T cells are separated and removed. They are genetically altered to have some specific receptors or special receptors called as chimeric antigen receptors or CARs. Chimera, as you remember, it was a fusion of different types of molecules or different entities, and this you can see in blue might have been the normal DNA of T cells of the patient, and the yellow one is what has been externally introduced, and that is leading to a recombined or new DNA, which is known as a chimeric DNA. This chimeric DNA is getting transcribed and translated and is being expressed on the surface of the T cells as a new antigen, which is a chimeric antigen, and it is being displayed on the surface as a T cell receptor. Now, what is done? Millions of such CAR T cells are grown and they are cultured, and then chemotherapy is given before CAR T cell therapy, and then the CAR T cells are introduced into the patient. And you can obviously understand that now, because you have a huge enriched population of a particular type of T cell, they are going to attack all of the cancer cells and they're going to help in clearance and they're going to supposed to give excellent outcomes. There are very many challenges associated with that also, but the overall concept is this, that you are going to modify or you're going to change um, the immune cells of a particular patient for better treatment. So let's go over the legend. The treatment principle of CAR T involves extracting the patient's own T cells using gene editing technology to transfect the chimeric antigen receptor gene, expanding the modified T cells in vitro as in culture in the lab, and then again infusing them back into the patient. The CAR structure consists of three distinct domains. We'll go into more details about this in the last slide, in the next slide. So, what are the three domains? You have an extracellular domain, a transmembrane domain, and an intracellular domain. The extracellular domain of CAR includes the antigen recognition domain, also known as single chain variable fragment, which is the SCFV, and the hinge region. The SCFV is composed of variable regions of both heavy and light chains of an antibody connected by a peptide linker, and CARs are being developed to target various tumor-associated antigens like CD19, 20, and 22. What did we mean? We'll come to that in the next slide. Just give me a second. Okay. So you can read this. We will actually go through the literature and simultaneously look at the schematic as well. First, focus on this first panel, which is figure A. So what does it show you? It shows you the structure and the mechanism of action of a chimeric antigen receptor. It has three domains. The first one is the extracellular domain, and it has that SCFV, which is a single chain variable fragment, and the hinge region. So this is your SCFV. What is its job? It basically means, what is an SCFV? Single chain variable fragment, which means that it is one continuous molecule. It's a single chain, but what it does is it mimics the structure of the two ends of the variable fragment. So for an antibody, so when was this? Yeah. So this, like you can see, you have two separate variable fragments. You have the heavy chain variable region and you have the light chain variable region, right? Together, these are forming the epitope, the paratope of the antibody where your antigen of interest can bind. So instead of having such a huge molecule, what this single chain variable fragment does is it just creates a binding site. It just creates its antigen binding region separately. You are synthesizing that region as a single chain. So this, you will have the first part, there'll be a linker, and then you'll have the second part. The rest of the antibody, the entire heavy load of the antibody is being eliminated completely. So that is exactly what forms the outer region or the extracellular domain of the chimeric antigen receptor. So this shown in purple replicates the two variable regions of the heavy and light chain. Along with that, Along with that, you have a hinge region. Hinge region is necessary for movement or for flexibility of the entire chimeric antigen receptor. Along with that, you have a transmembrane domain because, of course, it has to be expressed on the cell surface for it to be able to access all of the other proteins which it interacts with. And apart from that, within the cytosolic region, you have different domains. What are those domains? Let's come back to the legend again. So the extracellular domains include the SCFB and the hinge region. The transmembrane domain helps in anchoring the receptor onto the cell membrane, and the intracellular domain is actually responsible for signaling. The SCFB is engineered to recognize certain tumor associated antigens like CD19, 20, and 22. This region, this purple region. The hinge region allows for flexibility in the CAR's structure, while the transmembrane domain links the extracellular recognition capabilities to the intracellular signaling pathways. This domain typically contains co-stimulatory motifs that help in enhancing T cell activation upon antigen recognition. So this intracellular part, it has these two sort of motifs. One of them is for co-stimulatory. If you have studied immunology, you would already know this, that there are, apart from the main signaling or the main interaction between your antigen and antibody, there are co-stimulatory signaling that also help in boosting or enhancing the effect of that recognition. Usually it is between B7 and CD28, which are present on cell surface between the two cells. And these co-stimulatory molecules, they help in increasing the immune response, basically. That's what happens. So co-stimulatory molecules, they should have certain abilities which are necessary for the CAR to survive. And also in blue, you can see there are stimulatory molecules also. So these are just modifications that have come up over time, which I'll explain when we come to all these different generations of cars. So these are the basic ideas that have been kept in mind while designing chimeric antigen receptors. Now, what exactly happens under the therapeutic process of CARs? So, genetically engineered CAR T cells are infused into the patient where they're able to specifically recognize and bind to tumor antigens via their CARs. As you can understand, this is your cell um, like target cell, which is expressing this sorry, not target cell. This is your T cell, which has been infused back into the patient, and that patient is able to now express these chimeric antigens on their surface. Now, what is going to do? Genetically engineered cars, after infusion into the patient, they are able to recognize to and bind to the tumor cell antigens via these receptors. This leads to the activation of CAR T cells. Again, if you have studied the way in which T cell activation works, that once there is antigen antibody interaction and there is also support from the co-stimulatory molecules, it is going to lead to activation and then proliferation. Basically, there's a recognition of one antigen and one um, not antigen. There's an interaction between the T cell receptor and its cognate antigen. Then it leads to activation and selection and activation of that T cell. It increases a lot in number, and that logic is because this body has recognized that there is one particular pathogen and there is a need to clear that pathogen out of the system. So once that tumor cell antigen has been recognized, the T cell's job is to keep on expanding in number and then clear out the infection from the system. So this interaction leads to more and more activation of the CAR T cells and the release of cytotoxic molecules like perforin and granzyme B, which induce apoptosis in their tumor cells. So once that recognition has been actually facilitated, then what it does, it releases all these cytotoxic molecules like granzyme, perforin B, which help in creating pores within the tumor cell, and then the tumor cell dies. So that is the site. You are basically triggering the cytotoxic effect of the T cell by enriching the presence of tumor cell specific receptors on the T cell surface. One of the reasons why this needs to be done is because usually tumor cells or just cancer in general figures out ways of immuno evasion. So after growing within an individual's body, what they try to do is they try to avoid the immune system or evade the immune system. And therefore, a lot of times the tumor cell is not really able to recognize, the T cell is not really able to recognize the tumor cell as something foreign. It modifies itself in such a way that the recognition is compromised. So, what we are doing externally, or what healthcare workers are doing externally, is that they are helping the T cell recognize that tumor cell and clear it out of the system. That is the basic idea. Okay, so this is what happens conceptually. There are a lot of sub steps in this. We are not getting into all of that detail. If CAR cells are interesting to you, read more about not just CAR T cells, but CAR NK cells also, um, natural killer cells, because NK cells are more efficient at clearing out cancer. So they are being explored in that context also. But yeah, CAR T cells were the original idea. So it's more, you'll find more literature to help you understand that. So read more about it if it's interesting. But what I want you to understand finally, for at which we'll wrap up today's class, is the evolution of CAR T, how they have undergone modification over time. So, 5 generations of CAR T cell development have been actually recognized. So, the first generation CAR T cells had basic signaling domains, but there was no co-stimulatory signal. So, in vivo proliferation and clinical efficacy was quite compromised. So, like you can see here, only the only the single chain variable fragment was present, and there was a CD3 moiety which was necessary for stimulatory, but there was no co-stimulatory signal which could be elicited from such a CAR T. The same thing is shown here as well. So you have a CD3 zeta chain or the Fc epsilon R1 gamma intracellular domain without any additional co-stimulatory domain. So it had very limited capacity to actually trigger an immune reaction, or to even allow, like, even if recognition was occurring, like for example here between the tumor cell antigen and the CAR antigen, nothing was happening downstream of it because signaling could not be activated. So then over time, what happened in the second generation CAR T cells, there were additional co-stimulatory domains which helped in improving the potency of these signaling cascades, and there could be more persistence of that signal as well. Third generation led to further improvement where there was enhanced tumor lysis and cytokine secretion by incorporating dual co-stimulatory molecules. So here you can see more and more co-stimulatory molecules have been incorporated here as well. So yeah, you can see like CD3 zeta was already present. You have CD28, OX40 or CD2841BB, which is the example shown here. So this was leading to more enhanced tumor lysis and enhanced secretion of cytokines like interferon gamma, IL-2, TNF alpha, etc., etc. Then fourth generation CAR T cells were designed with controllable suicide genes and pro-inflammatory cytokines for enhanced solid tumor targeting. So they carried a nuclear factor of activated T cell, which is NFAT responsive cassette containing transgenic cytokines like IL-2. So they modified it even further in the fourth generation, where it had where it had these suicide genes, which led to even more enhanced effects. Let's keep it to that. And finally, the fifth generation CAR T cells, which is as updated as last year. I don't know about the exact current research because this is not really my field. But again, if it's interesting to you, you can look at it. But this is 2024. This is what has been uh, published in literature. That is the presence of the off-the-shelf universal CAR T cells, which is what I was explaining to you initially when I explained the domain of precision medicine. So these have been created by CRISPR-Cas9 gene editing to generate allogenic T cells addressing potential graft versus host disease issues. So additionally, they have also incorporated the JAK-STAT signaling pathway. They have also triggered the JAK-STAT signaling pathway, like here you can see, because they have added some IL-2 receptors, which will allow the docking of JAK and then the activation of STAT, and then STAT3 target genes will be phosphorylated, and then more and more activation can occur. So this is a whole other domain by itself. If we go into CAR T cell therapy, that will take like 3-4 classes by themselves, understanding the entire biology of chimeric antigen receptors. For now, just understand the concept of it, what you're doing, how you're using a patient's own cells, modifying it, putting it back into the patient, what sort of key modifications are necessary, what is the therapeutic effect coming from, and what are the modifications that have been made over time to improve it, and what is its practical applicability. So this is what we had to discuss for today. Think we're almost running out of time as well. Yeah. Okay, I'll just wrap it up quickly and then we'll take up questions. So basically today, what all have we discussed? We talked about small molecule inhibitors, which have tackled the challenge of undruggable TFs lacking a binding pocket. Alternative therapeutic strategies like PROTACs, protein degraders, and allosteric inhibitors have been actually used for targeted inhibition of mutant proteins. Precision medicine has revolutionized the field of cancer therapy, aiming at catering to unique individual specific differences. CAR T cell therapy is a cutting edge method of commercializing immunotherapy in clinics. And that sums up our key topics for today. Uh, the basic questions that I wanted you to address, first being the fact that what is the limitation of using PROTACs? Do they have off-target effects? I already mentioned it once before. Second, I would like you to figure out how much of the drugs we have discussed today. We've talked a lot about advanced cancer therapy, but how much of it is actually in clinical trials? So you can it's very accessible. You can just Google NCI clinical trials or NIH clinical trials, which is the National Institute of Health. And you'll figure out what sort of clinical trials are going out in the USA. You can check out if you can access this information for India as well. So that will give you a lot of idea about what drugs are actually being used or implemented for patients, how many patients are enrolled for a study, how many of them are given placebo, how many of them are given the actual drug, what are the parameters set, is age a factor, is any other condition required. So all of that information, it's if you have time on your hand, read about things like this. It's going to help you actually transfer what you're learning in slides and from books to actual patients, to individuals, real human beings. So all of that is pretty interesting. Um, one more thing I wanted you to look at is the idea of CRISPR-Cas gene editing. If you're not already familiar with it, this is something you should learn, because this is obviously, you can understand, this has received a Nobel Prize, so it's a quite a brilliant technique which has come in. But yeah, read about it. It's you definitely need to know it because other people are going to definitely learn about it. So if you're competing in some sort of an exam, this will it's not even going to give you an edge. You're expected to know about CRISPR-Cas in today's time. So read about it a little more. It's beyond the scope of this course, so I did not keep it here. It will take us away completely from gene editing. But yeah, if it's interesting to you, please look at it. And with this, I am done for now. If there are any questions, please let me know. We'll address that and then wrap up today's session. Okay, anybody else? Okay, if not, then I'll end the session for now. I might have gone by a little fast today, but yeah, I guess it should be. It was quite repetitive, a lot of things. But if there's anything that was unclear, you can bring it up in the next class. We'll discuss it. If not in the next, then in the last class. But yeah, that is all I wanted to discuss for today. I hope you learned something new. Um, there's always more to learn. So even if something you feel has left you with a few questions, open-ended questions, it's good to go back and actually read about it. Let me know if there's anything else I can help you with. And I guess, yeah, this is where we'll end our session for today. So thank you so much for joining. I will see you again on Thursday, that is on the 20th, from the same timing, 5 to 7 p.m. If there's any change in class timings, I'll let you know. But yeah, we'll have 4 more sessions, so please try to attend as many as possible. Uh, and yeah, that's all. So thank you. Have a good night, and thank you so much for joining.